Hi, everybody. I'm going to walk through doing the BioMan Mitosis mover exercise with you today. So let me start by sharing my screen. I'm going to try to make myself keep a good pace for us. Okay, so here in our Google Classroom, it's called BioMan Mitosis mover. If you open that up, it should bring you to a document here. It looks like this. On the left-hand side, it asks us to click here to access the website, and that'll bring us to the BioMan Mitosis mover simulation that'll help us answer all these questions. Okay, and it's been a little week for me or so this will be fresh for us. Okay, and I know I said in class, but as we click through, once you click, here I'm going to start a new game. It won't let me go back. So as we begin, make sure to read everything on the screen, and if not, that's why I'm here. Okay, so we'll come back here. Introduction. In order to make new body cells, the cell cycle must occur. During the cell cycle, one cell divides to form two daughter cells with exactly the same DNA. The cell cycle includes the following, and they give us 1, 2, 3 blanks. Here, interphase and mitosis. Here, that third one, cytokinesis, is the movement of cytoplasm at the end of mitosis. Here, they're calling it a third phase, but let's come back here. Interphase, the time between the phases when you're not, so just normal cell activity. Then there is mitosis, or cell division. And here they're considering cytokinesis as a third part, even though it is the end of mitosis, rather, the splitting. Next, interphase. Interphase happens blank, blank. Then just priming myself, we'll go back to our simulation. And here, the same intro. In order to make body cells, the cell cycle must occur. We already did this. Interphase, mitosis, cytokinesis. We see one cell splitting into two. You can hit the space bar or begin. Interphase happens before mitosis. That's what our first blanks are back here. Interphase happens before, that's goofy, before mitosis. Before the cell splits is when it's in the in-between time, the interphase. Next, it's asking during interphase, the cell does normal cell activities like making blank, for example. That it tells us here, normal activities like making proteins. Making proteins. If you make testosterone or melanin, whatever your cell is doing, you're making your proteins, normal cell activity. Number 3, uncoiled stringy DNA, I think is on our next one. Sorry, I'm not clearly. So here we're back to our thing. I'm going to click OK since we got our bit of information. Here we are beginning, I should say, before mitosis, we're in interphase. Let's make the nucleus and nucleolus inside the nucleus transparent so we can see what's going on. This is just us pretending to be able to see inside the nucleus. So here, interphase. Notice the stringy pieces of DNA in the nucleus. Uncoiled stringy DNA is called chromatin. That's our first blank. Uncoiled stringy DNA, chromatin. Let's stretch that out. The next question asks, the cell has blank pieces of chromatin. This means it has 2 blank molecules. I'm going to click next. I think that's what we'll find. No. The cell has 2 pieces of DNA. It's considering, or 2 pieces of chromatin. 2 pieces of chromatin. This means it has 2 DNA molecules. One wasn't spelled out as much. How many pieces of chromatin do each of your cells have? Me as a human, each of my cells have 23 pieces of chromatin, or 23 chromosomes, and then 2 versions of each. That's what we're going to see as we get into meiosis. Number 6, before mitosis can occur, the DNA must, and in parentheses it gives us the hint, copy itself. I wonder if the next one will give us that term. I'm sorry, I'm not being super clean with this. What is uncoiled stringy DNA called? This one, chromatin. We just got on the last slide, a chromatin. We're correct. I'm sorry, this one gives us that one we just filled in. This means it has 2 DNA molecules, 2 pieces of chromatin, 2 DNA molecules. Very simple cell. This one's calling us as having 46 pieces of chromatin. We have 23 chromosomes, which means we have 46 pieces of chromatin. And if that's what they're getting at with this, I will accept either because I don't know what you mean. 46 And then, sorry, now we're going to get to this. Before the cell can go, before mitosis can proceed, the DNA must replicate itself. Replicate is the word we're looking for in this last slide. DNA is going to replicate, it's going to copy itself. Okay. Sorry to be skipping back and forth. Okay, we're done. We can click OK. Click. Wait, oh, click on each piece of chromatin. Sorry, I did this with some of you. I have to work my cursor right over that piece of DNA. And as I click it, it duplicates itself. It's going to replicate that. And now we're ready to proceed. Okay, now I can hit OK. Which of the following are true statements? DNA replicates during interphase. So again, if we have interphase and then there's the mitosis, you want to copy your DNA before the cell is dividing. So yes, that is true. DNA is replicated during interphase. Chromatin is made of DNA. Yeah. Genes are found on DNA. Yes. We're barely getting into that, but yes. This one, let's hope I'm right. Yay, D. All the above are correct. Okay. Now mitosis can begin. Our DNA is replicated. Make sure I'm not getting over ahead of myself. After interphase, mitosis will begin. I think that's what they're getting at. After interphase, mitosis will begin. We can add that to our first blank. Mitosis. Mitosis is an important part of cell division, which allows you to make more cells. I think that's just taken from this. Allows you to make more cells. Making more cells is needed for growth, development, and tissue repair for healing from damage. Okay. We'll make sure we didn't miss any blanks. Making more cells is needed for growth, tissue repair. What was that middle one? We just read it. Development. I'm tired. Development. Oh, I can see why these text boxes are being goofy for you guys. That's okay. Mitosis results in the formation of blank, genetically blank daughter cells. The next slide will tell us. No. Intro to mitosis. So if you want to grow larger than a single-celled zygote, which is how you started, we need to make more skin cells or any type of body cells. We must do mitosis. We must divide. Okay, here's our answer. Mitosis ultimately results in the formation of two genetically identical daughter cells. One cell splits into two genetically identical. They are the same. We don't want mutations. We don't want things getting weird. We want to make copies of that same cell. The 4 phases of mitosis are same as written below. Prophase. What is that? PMAT. I hear it. Prophase, metaphase, anaphase, and telophase. And I've written them here. We can see them written below on this sheet. There's our prophase, metaphase, anaphase, telophase. Coming back to our simulation, I can guess what the next slide might show us. Nope, almost. Okay, mitosis. Why do organisms do mitosis? Let's see. A, to make more body cells like skin cells or liver cells, for example. Yeah. To make gametes like sperm or eggs. Oh, no. We're just beginning on that, but that's meiosis, where you want to make a cell that has half the number of chromosomes as the complete organism. So A is correct, B is not. A is mitosis, B is meiosis. So here, correct, A. And then here are 4 phases: prophase, metaphase, anaphase, telophase. I'm going to keep going. Mitosis, which of the following is the correct order for the phases of mitosis? Again, I'm thinking of PMAT for that PMAT. So starting with the ones that start with P, and then we can see C, prophase, metaphase, anaphase, telophase, PMAT. C. Okay. All right, now let's start mitosis beginning with prophase. Click OK when you are ready. Are we ready? I'm going to go back and make sure what we're looking for. Okay, so here we are on this sheet. That's my cat making noise. Prophase. One important event of prophase is that the chromosomes blank and become visible. Remember? It's going to show us. Condense. The chromosomes condense. They go from being a long strands of the stringy, thin chromatin into these much more visible. Okay, condense. Come back here. Prophase, they condense, become visible. And then next, the chromosomes have two halves. These are called blank blank. They are blank. That'll be in the next one. All right, let's click on the chromatin and make it condense. Ooh, there it goes. Ooh, and now that's what we're used to seeing as chromosomes, right? Tightly packed chromosome. Now we have two. Notice that each chromosome has two halves, and it's looking at, for that, the blue big G, that there's two halves of it, that this is a replicated DNA, two identical strands of DNA that are just attached in the middle. So these are our next blanks. These are called sister chromatids. The idea of that, like sisters, think of identical siblings perhaps. These are supposed to be twins of one another. So sister chromatids, identical, are our next blanks. Sister chromatid, those two chromosomes that are identical, replicated copies. They're sister chromatids, they are identical. And in the interest of your time, I'm not going to mess with these text boxes. Okay, to keep going. Sister chromatids are identical. In order to divide the DNA, we need to be able to move the chromosomes to opposite ends of the cell. Try to move the chromosomes to opposite ends of the cell by dragging and dropping them. So here, it's just me pulling one way, one the other way. Okay. Did you notice that the chromosomes are trapped in the nucleus? Again, we're seeing a cell with a membrane inside. That nucleus is the headquarters of the cell where the DNA is stored. Because of this, the nucleus disintegrates during prophase. So at the start, when we go from interphase and start mitosis, prophase, we dissolve the nucleus. Let's click on the nucleus to make it go away. Click. You can see it dissolve. And now we can manipulate those DNA. Okay. Come back here. Metaphase, to see what we're looking for. In metaphase, the chromosomes line up single file down the blank of the cell. In metaphase, the chromosomes line up single file down the middle of the cell. Middle, or the equator, is a term we've seen in other stuff. So here I'm just moving these chromosomes to that equator, to that midline of the cell. Okay. Sorry, before we answer the quiz questions, we'll come back here. You can say down the middle of the cell or down the equator. I'll come back to our questions. What happens during metaphase? A, chromosomes separate from one another. Chromosomes line up single file down the middle of the cell. Is B. C asks if chromosomes become visible. No, that's in prophase. So here, B. Again, meta means middle. So if you think about them lining up in the middle, whenever I see those X shapes, metaphase. Okay. Okay. So now coming back, we're going on to anaphase. So in here, we're looking for number 8. In anaphase, the blank blank split up. Here, the sister chromatids. Again, if this is the same chromosome, and we're pretending you got one copy from one parent, one copy from another parent, that DNA has duplicated. We're about to make two identical cells that will each have a full copy of each one of those chromosomes. So here I'm going to split up my sisters, dividing our identical chromosomes into two identical cells. Yay. So what was our blank? In anaphase, the sister chromatids. Those copied DNA strands. Now we're using a lot of terms interchangeably. Chromosomes, chromatids, DNA. Hopefully we're beginning to see that they all reference each other. Okay, come back to the questions. What happens in anaphase? The sister chromatids separate and move towards opposite poles. Chromosomes line up single file, down the middle of the cell. Middle means meta. Nuclei reform. That sounds like some endgame stuff. Nucleus disintegrates. No, that's at the start. So here, what happens in anaphase? A. The sister chromatids separate and move towards opposite poles. Or for me, anaphase, I think in the A and apart. They're being pulled apart in anaphase. This one's A. Okay, now we're in telophase. Telophase 2, diploid daughter cells are being formed. Daughter, the children cells, the resulting cells of this process. Let's see how it's accomplished. Or actually, sorry, let's go back here. I bet it's asking. In telophase, blank, blank, blank are being formed. 2 daughter cells. two daughter cells, two identical children cells are being created. And what we're looking for next on 10 and 11, each newly forming cell makes a blank. Let's see. I'm going to come back here. One thing that happens in telophase is that each newly forming cell makes a nucleus, which we'll see in a second. But let me come back here. Each newly forming cell will make a nucleus. Because again, we're at the end, we need to reform the nucleus that we took apart. And we're asking the chromosomes uncoil to make. I bet you could guess it. But here, we'll come back, click on the chromosomes to create nuclei for the two new forming cells. Oh, there's a nucleus. There's another nucleus. We got two new nuclei. Okay. And then here, the chromosomes uncoil. They condense to make the chromosomes. Now they're uncoiling back into chromatin. That's our last blank here with telophase, chromatin. Tin, I think of the tin as like tiny, tiny little strings. I don't know if that helps. We're almost there, guys. Okay. Uncoil my DNA. I'm clicking on it, goes back into the tiny strings of chromatin. Okay, we're ready to keep on with the telophase. What happens during telophase? Each newly forming cell gets a nucleus. Chromosomes uncoil to form chromatin. Two diploid. So again, full set of chromosomes, double number, not half number. Body cells are being formed. Hey, because we're just doing mitosis, normal cell division. We want a diploid full chromosome set as opposed to a haploid, which is our gametes, our egg and our sperm cells. Here, all of these describe telophase. Okay. All right, and then here we are in cytokinesis. I'm going to jump back to our document. It's asking, when does this occur? Well, after telophase. Usually at the same time as telophase occurs the cytokinesis. Click and drag the membrane to divide the cells completely. Well, I figured this is our membrane pinching. The cytoplasm needs to move before your membrane comes in. So it's a little bit of both. The cytoplasm moving and then pinching off. Okay. So at the time of telophase is when this occurs. At or after telophase. So we're going to put for cytokinesis here. And then it's going to get to our post questions in a moment. Right back here. Okay. Oh, we've made it. We've done it. Mitosis and cytokinesis. Again, mitosis being that cell division. Cytokinesis we consider generally part of that last phase, but the movement of that cytoplasm. And we've made two diploid, again, full set of chromosomes that are genetically identical. So let us see, after mitosis and cytokinesis are complete, which of the following are true? Do we get, first I would look at this of like, are we making 4 cells or are we making 2? Talking mitosis, this is normal cell division, we only want 2. We're just 1 cell split. So 2, should they be genetically different or genetically identical? Identical. Don't want going rogue, making little X-Men cells. Yeah, we want identical, no mutations, same cell being copied. Yay. Okay, let's do this. So here, if we are ending with 2 cells, that could be a great one to put towards the end. I know for me, we're starting with a cell that will have a nucleus intact and end with the creation of a nucleus. So this is where I would for me, that's where I begin, is to say, this is our end product. This looks like what we're beginning with. What do we think? Yes, I'm intentionally stalling on a video just because I want your brain to work, because I know it is. Here, they're going to line up in the middle before they're pulled apart to those separate poles, right? Here's our metaphase. Yeah, almost interphase. Then the nucleus would dissolve to make it prophase. Here's our metaphase as they line up in the middle. Anaphase, they're pulled apart. Telophase, those nuclei are forming around the 2 new baby cells, new daughter cells, and that cleavage furrow is beginning to pinch and form the new cell membrane after cytokinesis. You are done. You're a genius. Let's come back and do this last little bit. What is the result of mitosis and cytokinesis? 2 genetically or DNA-wise identical cells. We just made 2 new cells. We can answer these really quick. Why do cells complete mitosis? To grow, repair, replace older cells. Those are big ones. List the steps of mitosis in order: prophase, metaphase, anaphase, telophase. For the draw out, I will be happy if you just up in these little sections here on the right hand side, prophase, metaphase, anaphase, telophase, if you copy and paste some images from the internet, that will be sufficient. We want you to just get some good images that represent those phases. So I think for prophase, seeing that the chromatin is condensing into a chromosome. For metaphase, something that shows the chromosomes lined up in the middle. Anaphase, you'll see them pulled apart. Telophase, 2 cells coming from that. So we can see some 2 nuclei in that telophase. And any image showing that should be helpful. Okay, I hope that was helpful. I only took about 20 minutes. I'm going to stop myself. Please let me know if there's any questions. We can do this together another way. Thank you, guys.